So some of the most amazing things that are happening now, when you think about induced pluripotent stem cells, they're practically everything that people have done in genomics or cell biology can be done better with pluripotent stem cells because you can get them to reproduce indefinitely, so you can get huge numbers of cells. And you can turn them into every cell type in the body. So that means you can look at the transitions between an undifferentiated and differentiated cell. You can see how cells become neurons, how they become heart cells. And really at the basis, I think the most important thing is epigenomics. Epigenetics, what happens, how is gene expression controlled? And we know so little about that. And yet we have these stable genomes, or more or less stable genomes, and we can make all these different things using the only same 25 or so thousand genes. And development is one of the most astonishing things. When you think about in human development, in 9 months you go from a single cell to this incredibly complex organism. And all of that is through cellular interaction. And every one of those cells has to be making a certain suite of genes that causes it to have a certain identity, which is either the identity of the cell type that it is ultimately going to be, or maybe something just transient that influences some other cell, that influences some other cell to become the right thing. So when you think about human development, it is really epigenetics that is responsible for it. This incredible symphony of changes that take place so that genes go on and off, and they go on in concert and they turn off, and the right cell has to do the right thing. So it amazes me that we can even make human beings at all. I think induced pluripotent stem cells offer some unbelievable opportunities, and that's beyond the sort of obvious things. Cell therapy is what people mostly think of. And then people think about diseases in a dish, which I think is probably the second most interesting thing besides epigenetics. But being able to do really fundamental biology, this is like a perfect system. We can make leaps and bounds. So the second thing I think that's really important and in short term important is studying human disease. Because if you'll recall, it was maybe, I think it was about 30 years ago that the first mouse embryonic stem cells were made. And that led to the first mouse knockout animals, and knockout animals have led to the best possible models of human disease that ever existed. They've just completely taken over. Mouse models for cystic fibrosis and muscular dystrophy and Alzheimer's disease and every possible genetic disease you can think of. That's been what everybody's been working on. And now suddenly, we always knew the mouse wasn't perfect. I think people always have stories about curing diseases in mice and having no relevance whatsoever to humans. So suddenly this cell type comes along, which is essentially just as manipulable as a mouse, but it's a human cell. And now we can't make humans, but we can make these cells in a dish that have we can make all the cell types that make up a human. So if you're studying something like Alzheimer's disease, then you can study the very neurons that die in Alzheimer's disease. You can study the neurons that degenerate in Parkinson's disease. One of our projects is autism, which has been very, very difficult to work on in mice. You know, it's just mice with autism. It's very hard to find a model for that. But we have some genetic forms of autism that we're studying in the dish. We isolated skin cells from people with autism, and we've made induced pluripotent stem cells from those, and we've made neurons from those cells. And we can actually ask what's wrong with those neurons.